Hello everyone, welcome to today's live broadcast, Whole Genome, the future of high-quality genomes and transcriptomes. I am Judy O'Rourke of LabRoots and I'll be your moderator for today's event. Today's webcast is presented by labroots.com, the leading social media site for science professionals. Before we start, there are a few instructions. This is an educational webinar and thus offers free continuing education credits. After the webinar is over and to get your credits, click on the CE button located in the bottom left-hand corner of your web page. We want to hear from you during this interactive broadcast, so please ask questions or leave us a comment. Answers welcome too. You can do this by hitting the green Q&A button at the lower left of the presentation window and typing in your comments and questions. We'll try to get to as many as we can. Want a better look? You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you can't hear or see this presentation properly, let us know by clicking on the support button on the top right or use the Q&A button. We'll make sure we try to resolve any issues. Now let's get right to today's presenter. We are proud to welcome Jonas Korlach, PhD, Chief Scientific Officer for Pacific Biosciences. Dr. Korlach co-invented SMART technology with Stephen Turner, PhD, Pacific Biosciences founder and Chief Technology Officer, when the two were graduate students at Cornell University. Dr. Korlach joined Pacific Biosciences in 2004 as the company's 8th employee. He is the recipient of multiple grants, an inventor on 62 issued U.S. patents, and an author of numerous scientific studies on the principles and applications of SMART technology, including publications in Nature, Science, and PNAS. Dr. Korlach received both his PhD and his MS degrees in biochemistry, molecular and cell biology from Cornell, and MS and BA degrees in biological sciences from Humboldt University in Berlin, Germany. I will now turn it over to Dr. Korlach for his presentation.
Thank you very much, Judy, and also a warm welcome from me as well to today's seminar, Whole Genome, the Future of High-Quality Genomes and Transcriptomes, during which I'd like to give you some updates and some examples of the latest research from the scientific community applying PEC biotechnology and PEC biosequencing for higher quality genomes and transcriptomes. And the title, Whole Genome question mark, was inspired by a editorial perspective a few months ago in Nature Genetics, which had the same title, where the journal editors wrote that it is time, in their opinion, it is time to stop thinking that merely more DNA sequencing, by that they meant short-read DNA sequencing, will give us the variants that determine human traits. And they noted that our genomes come in many flavors of combinations of rare and common variation on many different spatial scales, not just single nucleotide changes, but also larger structure variation. And we know since 2007, this was the Craig Venter genome paper, that non-SNP DNA variation is very important and very abundant. In fact, the authors in this paper noted that even though non-SNP DNA variation only accounts for about 1/5 of all the events that were identified in the donor and Craig Venter, they involve 74% of all variant bases. That means 3/4 of all the bases that are different between you and me are not in SNPs, not in single base changes. And they suggested an important role for this non-SNP genetic alteration in defining the diploid genome structure. So what do we mean by non-SNP DNA variation? Up here is a graph showing the various kinds of so-called structure variation. That's larger variation where you have deletions, larger regions of the genome deleted in the individual compared to the reference genome, tandem duplications, variable number tandem repeats, dispersed duplications, novel insertions that only this individual has but are not in the reference, repeat insertions, inversions where the orientation is flipped, or translocations. And the importance of this type of variation, structural genetic variation, has been highlighted. This is just one example from a review paper by Richard Gibbs and Jim Lupsky from Baylor, noting that over 1/3 of all the genes in the human genome are encompassed either totally or partially by a copy number variation, another term for structure variation, that can alter their expression or even their structure possibly giving rise to novel fusion transcripts. And they note that detection of structural variation is imperative in any whole genome sequencing study. But of course, that's typically not being done with the short-read systems. And the limitations of Illumina sequencing or other short-read technologies has been highlighted in a number of papers. This is just one example from Stephen Scherer in Toronto, where they compared the different technologies, 4x4, Illumina, Solid, Complete Genomics, against the Human Genome Reference, and found that the current high-throughput short-read sequencing approaches only detected a fraction of the full size spectrum of insertions, deletions, and copy number variants compared to the reference, and they posited that generating longer reads can mitigate these shortcomings. So, as you know, smart sequencing has different characteristics from Illumina or even Sanger sequencing, and I've listed here the 4 performance areas that you want to consider really when evaluating any sequencing technology. And so you know that with the very long reads, the average are now over 10,000 bases, a high consensus accuracy of greater than 99.999% or QV50, or less than 1 error in 100,000 bases at 30-fold sequencing depth, the lack of bias with respect to GC percentage or low complexity sequence, and the unique ability to detect DNA modifications and characterize the epigenome as part of the sequencing process. With these performance metrics, it has now been possible to characterize regions and resolve genomes at a much more complete and comprehensive level. And so I want to give a couple examples of this in the following. The first example that I want to highlight is in the area of a class of diseases called repeat expansion diseases. And there are many of them. This is a review article from 2007 highlighting that the expansion of these short tetra, or more complex sequence motifs can lead to many different diseases, such as Huntington's disease, ALS, spinocerebellar ataxia, and so forth. And so, as just one example, this was a study that was published in 2014 by a group of Japanese researchers looking at spinocerebellar ataxia type 31. Now, in the human genome reference, you see up here on the top, there is a region on chromosome 16 that has 11 units of this particular pentanucleotide motif, TAAA. In an individual that's been that has the SCA31 disease, that's not what you find. Instead, you find this massive expansion and very complex pattern of these of all of these different repeats, which I've color-coded in the schematic in the middle, and then have listed them on the right. Moreover, so you know this is not part of the reference. Moreover, if you then look at the next individual, you see a very a similar but slightly different pattern. So if I move back and forth, you can see that here in the middle, this particular section right here is shifted. So the number of these repeat units are different. And so in the paper, it was highlighted that these are highly structurally dynamic, structurally unstable regions in the human genome, and that you really need long-read smart sequencing to resolve these. So this figure from the paper is 11 patients that were looked at with smart sequencing. The canonical sequence on the bottom was determined, and then the bars in the graph represent the length of these particular repeat units, so the K, L, M, and N. And you can see that for each of those patients, they're all different, highlighting the high structural diversity and variation in these repeats. The paper also pointed out that there are many regions in the genome that are longer than what you can find in the reference genome. So in the figure here on the bottom, you see that it might be possible to resolve some of these in the reference, but then precisely for the individuals where you're trying to make a diagnosis that have the disease, you run into the limitations of the short-read sequencing technology. So probably around this area right here is where the limit for the short-read sequencers are located. That's about 200, 250 bases. So some of these repeats stretch out to 600, 800 bases, and you really need an unbiased long-read technology that can go through these types of repeats to resolve them. And this is not an isolated example. I just want to mention one other paper looking at another type of this spinocerebellar ataxia, type 10, and a very similar picture emerges. Here the figure on the right for patients A, B, C, D. You see this very large heterogeneity and diversity in the structural composition of the disease implicated repeats. The second example that I want to highlight for resolving structure variation and complex regions in the genome in a targeted fashion is in the area of pharmacogenomics. Cytochrome P450 2D6, or CYP2D6 as I call it, is a very important gene. It's directly involved in the metabolism of about a quarter of all the commonly used drugs. It's a clinical target for drug metabolism efficiency. And it's difficult to study because it's a highly polymorphic gene. There are over 100 variants, including a full gene duplication and deletion events. And so most genotyping assays, because of this complexity, only interrogate a small subset of the variants. And they default to the diplotype standard call in case this information is missing in the panel. So just to highlight the importance, so different people have different genotypes, and that directly affects their phenotype on how quickly and how efficiently they can metabolize these drugs. So in order to do this research and ultimately have a better understanding of how much of the drug should a particular patient receive, either during clinical trial or then ultimately in the future for patient management, it's very important to know this genotype-phenotype interaction. And so Scott Stewart at Mount Sinai has published a paper and presented this work, highlighting that it's complicated by the fact that there is a copy. There's a neighboring pseudogene which has 97% homology right next to the CYP2D6 gene. And in a paper he published a little while ago, Stuart Scott, entitled Long-Read Single Molecule Real-Time Full Gene Sequencing of CYP2D6, he pointed out that short-read sequencing is not an effective approach for CYP2D6 full gene characterization, and that long-read CYP2D6 smart sequencing is an innovative, reproducible, and validated method for characterizing the full gene, identifying the duplications, and discovering novel alleles, which will likely improve the CYP2D6 metabolizer phenotype prediction for both research and the clinical testing applications. So just a couple examples. He initially validated the method by looking at 10 previously characterized DNA samples. They had perfect concordance with the previous genotype calls. And in fact, one of the samples showed a duplication events that had been missed by the other platform, and the genotypes were refined in some cases. So here you see that there was refinement of the genotype from the previous Luminex or TACMAN copy number assays. This is the diplotype from smart sequencing. There was a duplication allele that was characterized that was missed before, and there were a number of novel alleles that were characterized. And then he looked at 14 samples with discrepant results from the multiple genotyping, the Luminex and the TACMAN, and were able to resolve those fully with suballele resolution, refining the genotype, characterizing duplications, and in some cases finding completely novel tenomer rearrangements that were not found before. So then to address the question of, well, how much structure variation is there in the human genome, this was a paper from last year looking at and trying to address this question. Evan Eichler and colleagues looked at a particular human DNA sample and applied smart sequencing. And so in this study, they resolved over 26,000 of these types of structure variants at the base pair level. And remarkably, 85% of these, about 22,000, were completely novel, highlighting that we've really had a blind spot in the area of genomics, not being able to resolve and understand and capture those particular types of genetic variation. Almost 7,000 of these were inside genes, and the validation rate was really high. So in the paper, you can see this figure that it spans, this type of variation spans all the way from 50 bases all the way to 20,000 bases. And all the data that you see in black and red were all new discoveries, and in gray and pink were previously reported structure variants. And some particular classes are particularly poorly represented through the short read sequencing technology. So here's an example where less than 1% of insertions are present in the newer assembly that were derived primarily through Illumina sequencing. So here is complex insertions, short tandem repeats, and variable number of tandem repeats. The PacBio work identified about 1,700 events, adding about 3.5 megabases of sequence, and the same sample sequenced with Illumina only identified 14 out of those 1,700, and you can see that they're also very poorly represented in the reference. The latest reference only has 5 of these events capturing this particular type of variation. So to summarize, this is a very famous graph, of course, that I'm sure you all have seen, where that talks about the decrease in the cost of sequencing. But what I want to highlight is there's a dilemma of the quantity versus the quality of the information that we're getting. Yes, sequencing has gotten very cheap. However, the ability to comprehensively characterize the genome and to assemble human genomes de novo certainly has not followed that same trend. And in fact, according to the NHGR website, the very definition of what it means to sequence a genome was actually changed in 2008 from an assembly viewpoint to a remapping viewpoint. And so I just want to play a perspective that David Hausler from the University of California in Santa Cruz gave on this particular issue. So then I want to now talk about de novo assembly, and this was nicely highlighted, the need for being able to put together human genome from scratch was highlighted nicely in this review article, Genetic Variation and de novo Assembly of Human Genomes by Mark Chase and Rick Wilson and Evan Eichler, where they pointed out that the short read massively parallel sequencing is insufficient to generate high quality genome assemblies or resolve most structure variation, and they emphasized the importance of complete de novo assembly as opposed to read mapping as the primary means to understanding the full range of human genetic variation. So there has been a transformation through the introduction of the smart sequencing technology in our ability to put together human genomes from scratch to higher quality. In this graph, I show a timeline. You see on the left here the year when these studies were carried out, and this goes from 2007 all the way to last year, looking at the different technologies with different assemblers. And then the graph on the right shows one metric of the quality of a human assembly, which is CONTIG N50, and that's a metric of the contiguity. And you can see that over the last 7 years, between 2007 and 2014, really the community hasn't made much progress on being able to generate high-quality de novo assemblies. And with the emergence of the PacBio RS2 and smart sequencing and the Falcon assembler, it's now possible to generate very high-quality human genome assemblies that have CONTIG N50s in the megabase range. What that means in practice is shown here, where the latest assembly of CHM1 just showing 2 chromosomes, where in blue you can see individual CONTIGs that are greater than 5 megabases, and so some of these CONTIGs span entire chromosome arms in an uninterrupted fashion. These are CONTIGs, not scaffolds, so they only contain A, G, C's, and T's, and no N bases, no gaps, and completely resolving this. This has been described in other papers. This was a paper by Adam Filippi and colleagues developing efficient assembly algorithms to give rise to these higher quality genomes. And on the figure on the right, you see a comparison. On the left of each chromosome arm is the PacBio assembly, and on the right of each chromosome arm is the corresponding short read assembly. And then in terms of the ability to see the genome comprehensively, this is a study led by researchers at Mount Sinai, a de novo assembly of a human genome. And on the bottom here, you see again the ability to see lots of structure variants with the PacBio. And then in contrast with the Illumina technology, you only see a very small fraction of this type of variation. And this paper also pointed out that certain types of variation are systematically underestimated in the human reference genome. So this figure is particularly about short tandem repeats, where compared to the reference, you have an overrepresentation of data points indicating that these are much longer than they're actually present in the reference genome. So I want to give one example of what it means to have a higher quality reference. And what I'd like to focus on is the immunoglobulin heavy chain locus, the IgH locus, which of course is extremely important because it's responsible for antibody heavy chain biosynthesis. It's vital for the adaptive immune response, so it's naturally crucial for understanding many diseases. And in addition to the reference effort of the human genome reference, in 2013, a paper was published by Rick Wilson, Evan Eichler, and others with a very laborious and time and labor-intensive approach to resolve a second individual. This was a hydatidiform mole, to get a second haplotype representation of this particular locus using a back-based approach and using Sanger sequencing. And as you can see here in the figure on the top, you can appreciate why it's so difficult to resolve these particular regions that include the VDJ regions that make then the antibodies, because they're highly repetitive and very complex. Having a high quality de novo assembled genome with PacBio can give you these regions automatically, just straight out of the assembly. So for two hydatidiform moles, CHM1, that's the same one that was used in 2013. And then another one, CHM13, if you look at the assembly and ask where the IgH locus is, they're fully resolved in single large CONTIGS in the PacBio de novo assemblies. In CHM1, that region lies in a CONTIG of 6.6 megabases, in CHM13 in a CONTIG of 1.7 megabases. And so you can then immediately ask about the differences, and so the bioinformaticians don't have to spend all their time trying to put things together, but they can actually now go to what they wanted to do in the first place, which is to analyze and annotate those particular regions functionally. And so for CHM13, I'd show by example, this is a dot plot, which gives you a high-level comparison between the sample of interest of that particular region and the reference sequences on the y-axis. So this is a large region, about 1.4 megabases depicted here. And when you have a diagonal line, that means the sequences are concordant. When you have breaks in these lines, that means there are structural differences. So it's a little bit hard to see on the screen, but there's a discontinuity here, and because it's a large scale, so I zoom into this particular region. Here is a short tandem repeat expansion where you have 2,350 base pairs in the CHM1 sample versus 50 bases in the reference. Here's another example, 2.6 kb of short tandem repeats compared to 760 bases in the reference. There is a 19.5 kilobase tandem repeat expansion in this IgH locus in CHM13 compared to the reference. There's an inversion of about 5 kilobases, which you can identify in the dot plot like this. There's an 8.5 kilobase insertion at this locus, another 25 kb tandem repeat expansion here, and another short tandem repeat expansion there. So you see that there are quite a number of structural differences, large structural changes between the particular sample that was sequenced and automatically gave a complete picture of the IgH locus compared to the reference. And then we can compare both of the samples, CHM1, CHM13, and you can see that some of the variation is the same between the two. So for example, this short tandem repeat expansion, the inversion is the same, this particular insertion and so forth, and some are unique. So for example, here in CHM13, we had the 25 kb tandem repeat expansion, which is missing in, and this is concordant with the reference. So it gives you an immediate view. Now, how did the short read assembly compared to this work. There was an Illumina-based, not a de novo assembly, but a reference guided assembly done before the PacBio work, which also, by the way, included a large number of backs. The CONTIG N50, as I mentioned before, is only about 140 kilobases. And in this assembly, the IGH locus is broken into 3 pieces in a scaffold. And it has 2 very large gaps. One is 1 kilobase, and the other is 50,000 bases, 50 kilobases. And the IGH locus is not correctly resolved. So anywhere you go in this particular region, you have the letter N's instead of actual AGCT bases. And then you can ask, well, how did the PacBio assembly resolve those particular regions? The first gap was filled with 2.8 kilobase of actual real sequence, and the second gap was filled, it's not 50,000 bases in length, but it's actually 135,000 bases in length. So even the estimate of the size of the gap was incorrect in the short read assembly. So to summarize, there's a substantial difference between when you talk about contigs and scaffolds. Contigs are continuous pieces of sequence information that contains only ACGs or Ts, and that is what PacBio technology provides. Whereas scaffolds, some researchers have called them Swiss cheese genomes, have lots of holes with a varying number and size of these gaps of N bases. Those are holes in the genome which translate to missing information and gaps of knowledge. And one of the, read length is not enough, so you need read length to span the repeats and to resolve those into the contigs. The other aspect is the lack of GC bias, and this was also nicely highlighted in the paper, the difference between the 2 different technologies, where you have the Illumina sequencing, and it's well known that at low GC content, so down here, this is AT-rich DNA, and then high GC DNA, there's bias, and there's much less coverage and lower quality sequence information, whereas with the PacBio technology, which is shown here in green, you have a very even coverage and quality regardless of the DNA that you're trying to sequence, and that is a fundamental prerequisite of being able to characterize genomes comprehensively. The last thing I want to note about human genome reference assemblies and de novo assemblies is the importance and the process of what we're seeing now of the generation of ethnic background sequences, ethnic specific de novo assemblies and human genome references with the use of PacBio technology. And that is important because the ethnic background critically matters to disease risk, disease association. I just want to highlight 2 examples. This first one is for the disease rheumatoid arthritis. These Korean researchers looked at a set of SNPs that were associated in a Caucasian population to contribute significantly to disease for rheumatoid arthritis. And they asked a simple question of how many of these SNPs play a role in contributing to the disease in the Korean population, and the answer was 0. So none of the SNPs that were identified in the Caucasian cohort had any meaningful contribution to the Korean population. This, of course, is also important in cancer. This is a very nice review paper looking at the role of ethnic background and genetic variation in the evaluation of cancer risk. And the authors here surveyed a number of studies and found that a significant result in one ethnic group was usually not reproducible in other ethnicities and well-powered studies. So that is the reason why now the community has engaged in generating high-quality reference genome assemblies with PacBio technology. Here's a table of the publicly announced projects, and so you can see that there is quite a wide variety of Asian, European, African genomes that will give us, that are projected to give us a much better match of the genomic data that you get from population studies to the particular reference that matches much better the ethnicity of the cohort. The last example of a recent PacBio genome assembly that I would like to mention is the gorilla genome. This was featured on the cover of Science last month, and it provided a dramatic improvement over the previous gorilla assembly. Of course, the gorilla is not only important for conservation efforts, but also is our closest relative phylogenetically. And so for areas like understanding speech, neurological conditions, many diseases, it is, and for comparative genomics, it's very useful to have a high-quality gorilla genome. And so in this particular PacBio assembly, you can see that there was a dramatic improvement in terms of the contiguity, over 150-fold. Many of the gaps, over 93% of the gaps in the previous assembly, which was in scaffolds, not in CONTIGS, were closed. It added 150 megabases of new sequence, corrected errors. Similarly, about 86% of the structure variants are completely new, and it had corrections to our previous estimates in terms of evolution. The paper had some really nice figures that I think highlight the difference between the PacBio assembly on the left, so this was from the new work, compared to the previous short read assembly with regard to what you're looking at as a scientist when you look at long read versus short read assemblies. This is a figure of in the paper showing a histogram of this information of the contig length, and you can see in black and in blue the previous Illumina and Sanger sequencing contigs, and then in red the PacBio sequencing context. And this is a log scale, and so you can see that the distributions are completely separate, and there is a 1,000-fold difference and improvement with the PacBio technology. So Evan Eichler, the senior author of this paper, was interviewed for a blog post accompanying this paper, and he had some, I thought, really nice statements. So I apologize, it's going to be a little bit of text, but I couldn't really say it any better than he did. And so he noted that the short read assemblies can be like Swiss cheese with lots of missing biological information in the gaps. The gaps are not random and are clustered at sites of repeats. And if the geneticist can't capture these repeats and determine the structural differences, they have problems understanding the organization of genes and comparing genetic variation within and across species. He noted that through PacBio sequencing, the technology and the computational biology have now advanced to the stage where individual laboratories can generate high-quality genomes of mammals, and this capability has the promise to revolutionize our understanding of genome evolution and species biology. He also pointed out that he thought the take-home message from this study was that the new genome technologies and assemblies bring us back to the place where we should have been 10 years ago. So coming back to that graph and the comment that David Hausler made with regard to quantity versus quality. He said that he thinks that if we depend only on short read sequences, there's a chink in our armor. The work on the gorilla and other human genomes clearly demonstrates that large swaths of genetic variation can't be understood with the short read sequencing approaches, and long read sequencing is allowing us to access new levels of genetic variation that were previously inaccessible. And he concluded that given further advances of the technology, and I'll talk about the sequel system at the end of the presentation, I'm willing to bet that this is the way we will sequence human genomes to discover disease-causing mutations in the future. So beyond the human and our closest relatives, there is quite a number of projects that are now underway and have already been published on applying PacBio sequencing to get much higher quality genomes of plants, animals, bacteria, and basically all parts of the tree of life. This is not meant to be a phylogenetically accurate tree, of course. It's just a illustration that this is now happening. And these are all the genomes that we know of that have a contig N50 of greater than 1 megabase. And there's a hashtag with the club. If you have one, you can also participate. So then, in the last sort of 10-15 minutes, I want to now switch to the transcriptome. Of course, both all the genome, transcriptome, and also the epigenome. We won't have time to talk about the epigenome today. But the transcriptome, of course, is just as important in understanding biology, understanding disease, and the molecular mechanisms in addition to the genome, because it's up to the gene products and the regulation that are the underlying foundations of many of diseases. And so it's been recognized, of course, that a particular gene can have many different gene products. And for example, in the human genome, it is estimated that over 95% of all human genes are now alternatively spliced, meaning they have more than one isoform, more than one form of what the gene can represent. And of course, this is done through alternative splicing of the introns and so forth. And it's been well recognized in the community that the short-read sequencing technologies have difficulties resolving this architecture, this complexity. And if you are limited to the reads that just happen to span the splice junctions, there is insufficient connectivity, and so that leads to the splice isoform uncertainty. We and others have developed a solution that we call isoform sequencing, or ISO-SEQ for short, where the cDNA libraries that are being made encompass full-length cDNA. So the entire mRNA molecule is converted into a corresponding cDNA molecule, and then with the long reads, that is sequenced with full resolution and with full contiguity. So that means there's no assembly required at the end, and you get the isoforms out automatically out of the sequencing process. There are many publications that have now appeared. I've listed a few of them here, and I understand we're making available the presentations PDF, so you can look this up. There's also a library resource on our website where you can get to these papers. So I want to just highlight a few. So we talked about the IGH locus earlier, and there are a number of papers that highlight that now on the mRNA level, when you make those heavy and light chain RNA molecules that then make the antibodies, of course, it is now very easy to read through and directly resolve the BDJ recombination events and characterize the immune repertoire, either in animals, and these are two studies that are in animals. On the right here, you see in the figure on the lower right here, you see from researchers at the USDA, Tim Smith, looking at the bovine IgG repertoire. Of course, this is very important for disease susceptibility of calves during transport, for example. And you can already visually appreciate that through this characterization, there's some very different constitutions in the immune repertoire in some of these different animals. The ability to read mRNA molecules full length is very useful for characterizing fusion genes, and of course they are particularly important in cancer. Here's just one example of a number of papers that have appeared now. This is in prostate cancer, a novel fusion transcript between the human relaxin 1 and the human relaxin 2 gene. This paper, I think, is a nice example because the authors highlight that they basically, through the kitchen sink of sequencing technologies, added and couldn't identify this fusion until they used PacBio sequencing, which very confidently identify that there is indeed a fusion between those two genes, which have a high homology. And so it's quite difficult with short-read technologies to resolve this. And in terms of the functional significance, just one example. So they identified that one of the fusion encodes an isoform that does not then have the signal peptide. And so without the signal peptide that normally would dictate the transport through the Golgi apparatus and determine the destiny of that particular protein inside the cell, this particular fusion protein will end up in a completely different cellular compartment because it lacks the signal peptide. So having this full contiguous information really then helps you go directly to the next level of understanding it functionally. And just one other example in the area of cancer, looking at alternative splicing. This is from a presentation that was given at the American Association of Cancer Research in New Orleans last month by researchers from the Netherlands Cancer Institute. They're interested in looking for novel biomarkers for colorectal cancer. And it turns out that as you go through the progressions in colorectal cancer from the adenoma to the carcinoma, there are changes in the RNA splicing in many genes in the tumor. And so looking at differently spliced isoforms can give you an understanding where the cancer is at. and could complement or outperform the current screening to give information about these particular cancers. So a couple of examples looking at two samples. The green sample is the control sample. This is a cell line. And then in purple is a cell line where the aberration or the changes in the splicing machinery have been mimicked through siRNA mediated downregulation of components of the splicing machinery. And so you can see that through ISO-SEQ and the collection of full-length mRNA isoforms, it's very easy to see the changes. These are data that were done on the SQL system. And so you see that for this particular gene, there's only one isoform in the control, and then there are three gene products in the cell line that has the modulation of the splicing machinery. And you can see which particular exons are being spliced out. Here's another example. And this is a gene that when we found this and communicated this to the collaborators, they said, oh, that's one of the genes that that's in our top 5 list. So they asked me to block out the identity of the gene because that's a gene that's very important to them. But you can see that compared to the control, there are now changes and exon splicing again, and you can immediately identify which of the isoforms have the exons that are spliced out and which are still in the normal state. So lastly, looking at the transcriptome and how much do we know already about the transcriptome versus how much is there still to be learned, there was a paper in 2013, one of the first ISO-SEQ papers, looking at what is arguably the best studied transcriptome in the world. That's human embryonic stem cells. So literally billions of reads have been, billions of Illumina reads have been spent on that sample. And in this paper, the authors applied PacBio sequencing and found that over 1/3 of the reads that came off the machine were either new isoforms or even new genes. So they identified 273 genes and concluded that even in these well-characterized human cell lines and tissues, the mere process of identifying all the genes is likely far too complete. And they have since gone on and interrogated these discoveries further and found that some of those new genes were crucial for regulating pluripotency during human pre-implantation development and nuclear reprogramming, so during development. So the ability of PacBio sequencing to make these discoveries then immediately led to the identification of these crucial regulators. And this was very nicely highlighted in a technology feature in Nature a few months ago that I recommend if anyone is interested, entitled Finding Function in Mystery Transcripts. So these were directly enabled by PacBio sequencing data discoveries. New transcripts also help you make better sense of the short read data. So coming back to the PNAS paper, they showed that there is a much better quantification with the inclusion of the new transcripts. You basically have a better transcriptome reference, which then allows you to make much more accurate calls of the short read data for gene expression quantitation. And then this was I think, an exciting first paper in going to the next level of looking at what we would call personal transcriptomes. So of course, you all have 2 copies, except for the male sex chromosomes. Everyone has 2 copies of the same gene, and those can be differentially expressed, and in some cases differentially spliced. And so Mike Snyder and co-workers in this PNAS paper utilized full-length ISO-SEQ PacBio sequencing to differentiate the transcripts from the different alleles. So in this figure here, you see that for this particular gene, they were able to cluster all the transcripts that came from the first allele using SNP differences that are shown here by the orange lines here and there. That allows you to cluster and identify which allele, whether that particular RNA came from the mother chromosome or from the father chromosome. And you can see that it matters for some genes. So for example, here for all the transcripts, or virtually all the transcripts that are expressed from allele number 1, this particular exon is spliced out, whereas from the other allele, half of the time, this exon is retained. So it matters which allele that particular gene product comes from, and with regard what's going to happen to it. So the last example in the transcriptome is in a plant area, and this is a work by Bo Wang and co-workers from Doreen Ware's lab at Cold Spring Harbor Laboratory, looking at maize. So corn, of course, is the largest crop, and I didn't have time to talk about the genome, but they also now is available publicly the latest version 4 reference sequence, which was done with PacBio sequencing. And Doreen mentioned in her talk that previously the maize project was a $30 million effort by the USDA, led by the USDA with many other institutions that led to the version 3. And now with PacBio sequencing and a estimated cost reduction of about a factor of 1,000 to about $30,000, which was done at the time, now it would be cheaper still. They have a version 4, which has a much higher contiguity. I want to focus here on the transcriptome part. And of course, it's also important for breeding efforts and for understanding the different varieties and genotype-phenotype correlations to get an idea about the transcriptome. So different tissues were collected and subjected to the ISO-SEQ protocol, getting full-length cDNA that was sequenced in different size ranges. And there was also a multiplex strategy involved where the different tissues were barcoded so that this can all be done in one library preparation. And so you can see here that the stark difference in the availability of transcripts that was possible in blue with the previous version 3 reference and then with the ISO-SEQ data. So you can see that there's a large fraction of new transcripts that are in the range above 2.5 kb or so that are newly discovered. And this is also shown here in terms of the number of different isoforms in genes. There is a great discovery potential of these new isoforms and sometimes even new genes. So in fact, about 60% of all the data were novel isoforms either from known genes or from novel genes. So you can see here the blue one was already known, and then in red and in red here, 57% are novel isoforms from known genes, and then 3% were novel isoforms from novel genes. And so this allowed, for example, just as one example, allowed a much better characterization of the functional diversity of isoforms in 57 maize transcription factor families. And what's shown here in blue are new discoveries. So you can see that for all these different classes of transcription factors, there is a very large component of new discoveries that was made from the PacBio data. And so it then allowed Bo to get a much more comprehensive and complete picture about tissue-specific transcripts and characterizing the alternative splice events among the different tissues. He found that in some cases there's quite a lot of mutually exclusive exons and intron retention going on. So for example, for these two, you can see that the turquoise bars are for mutually exclusive exons, make up a very large fraction in the endosperm and the pollen. And here's just one example of a retained intron that can then, of course, cause a change in the ORF and can have different functional consequences. And so this is only possible if you have the full-length information to understand this diversity. And these were also validated. So, and then previously incorrect gene models have now also been validated by the ISO-SEQ method. So here are two examples of genes which were incorrectly annotated in the ref gene version 3. So here is the annotation on the version 3 reference, and so it's kind of split. Whereas by looking at the different tissues with PacBio here, it's very clear that these are continuous transcripts, and then down here you have the different tissue distributions and the different isoforms. On the bottom here is a gene that was completely missing, so you can see there's actually no line that corresponds to the 3 reference, and very clearly 2 different isoforms were identified in root and tassel. Also, it allowed them to characterize really well the novel long non-coding RNA or link RNA. So over 800 novel link RNAs were identified in this particular work. And you can see here's the breakdown between known and novel link RNAs, giving an insight into the particular constitution. And then here on the lower right, you can see that many of these link RNAs are very tissue specific. So there's very little overlap in with regard to link RNAs that are present in all the tissues, and a large number that are present in only one particular tissue, potentially giving insight into the particular regulation in these tissues. And so, speaking of long non-coding RNAs, there is an exciting abstract. I hope you can read this at the upcoming ASHG meeting that's in about 2 weeks in Europe. This was a group from Barcelona, Spain, looking at mapping human long non-coding RNAs. And just to read, this is from the abstract, so I'm looking forward to this presentation, where the application of third-generation long-read sequencing in both human and mouse led to a set of canonical link RNA splice junctions that increased by 250%, so it more than doubled the number of known link RNAs in these gene models. They observed numerous cases of new exons, unified gene models underlying multiple annotations, and monoexonic genes. And they also mapped 132,000 polyadenylation sites genome-wide. So with regard to link RNAs, enormous discovery potential as well as for in the agriculture business. So with that, I'd like to conclude by saying that I hope I've given you an overview that there's a lot more information that can be gathered from the genome, more than just SNPs. And of course, in that context, the Illumina technology is very good at identifying, for the most part, for most regions of the genome, the single nucleotide variation, but it largely ignores the larger structure variation, the phasing of the long-range phasing of the haplotypes. In many cases, because of GC bias, there's the need for laborious gap filling, and then also epigenetics and de novo assembly. And while it is true that it can be, at least in many cases, partially addressed with more made-pair libraries, with these synthetic read technologies like Dovetail, 10x Genomics, gaps can be addressed with PCR cloning, Sanger, epigenetics with bisulfite sequencing, and de novo genome assembly, again with cross-linking methods like Dovetail and Hi-C. This adds a lot of work and certainly gets you far beyond the so-called $1,000 genome, which is really just a SNP table against a reference genome and does not give you all this information. And so the point I was trying to make, both in a targeted, in a resequencing, and then in a de novo assembly manner, is that smart sequencing can give you a data set, a homogeneous data set from a single library that can provide all this particular information. And often the gap filling, goes away because there's no need, because there are no gaps in the CONTIGS rather than having scaffolds. With regard to the RNA level, we find that in the community that in many cases the researchers combine short read and long read sequencing, and so the technologies are complementary in that regard, because Illumina can still give you a lot of counts and billions of reads, so that's very good for counting. We don't have those read numbers yet, but as I mentioned and showed a couple of examples, the short read technologies are perhaps not as good as highlighting and resolving and capturing the transcript structure, and that's where ISO-SEQ is really good at. So being able to use ISO-SEQ for characterizing the isoforms and the transcript structure, and then complementing that then with the counting information is, we believe right now, the best way to give you comprehensive information on the transcriptome. So with that, I just want to make a few remarks about the SQL system. You may have heard this. This is the instrument that we announced in October, and it gives you a higher throughput on the existing smart technology. So the underlying technology is exactly the same, but we've now increased the capacity of the number of potential sequencing sites per smart cell to 1 million, and that relates to about a 6- to 7-fold increase in the number of reads that you get per run. It also has a decreased footprint and weight, and is by about a factor of a third, respectively, and is about half the cost of the RS2. And so what that means is that with the larger number of reads, it reduces the time to the results and the time to do a study. Importantly, it also has an inherently scalable throughput, and I want to just make one comment on this, that in contrast to the RS2, where we really hit a limit in terms of how many sequencing sites we could have in a SMRT cell, this is not the case with SQL. So over time, it will be possible on the same hardware instrumentation to upgrade the SMRT cells to giving higher multiplex to 4, maybe 8 million ZMWs. And so on the existing instrument, it will then give you even more reads. And so that means that over time, the cost that it will take to do a human genome de novo assembly, about 50-fold coverage is our recommendation, from the current value in terms of U.S. consumable costs, will go down from currently about $17,000 over time in 2 years or so to a price point where it's then possible to do more and more samples for larger and larger population-based studies. So with that, I would like to conclude, but not before a few acknowledgments. And this is actually my first acknowledgement slide, which is to highlight that the use of the technology by the scientific community has now led to over 1,300 papers, to well over 1,000 peer-reviewed publications, and there are already about 350 or so papers just this year. This is an approximate number because it's actually starting to get difficult to track the papers precisely because they're coming out at such a rapid rate. We have between 25 to 30 papers each week that publish on the use of smart sequencing in the various different areas, or that translates to one new paper on average every 6 hours. So it's getting hard to keep track and read them all, but that's a really good problem to have. And so I show this slide because it is really the research community and the researchers and scientists who apply the technology that we have to thank for, and we are extremely grateful for their time and efforts that they put into the technology. And without them, this would not be possible. It's a very close collaboration. As you noticed, most of the things that I talked about were actually not done by us, but were done by the scientists in the community. And of course, I also want to thank all the employees at Pacific Biosciences who work very hard every day to bring this technology to your research. So with that, I'll stop, and I'd be happy to answer any questions.
Excellent presentation, Dr. Korlach. Thanks for bringing that information to us. So before we get started with all of your questions, here's a quick reminder about how to reach us today. Questions can be sent via the green Q&A button at the lower left of the presentation window. We'll get to as many as we can. And our first question is, what is an isoform?
So an isoform is a particular form of a gene product. So when you have a gene in the genome, it's made up of usually more than one exon. So it's made of exons and introns. And in the process of transcription that makes the messenger RNA and then is processed by the ribosome to make a protein, the introns are spliced out and the exons are stitched together. And sometimes, an exon is skipped, and sometimes an intron is retained. And so with this modular architecture of a gene, you can end up with different messenger RNAs that are then processed by the ribosomes to make different proteins.
How does this technology work in the single cell arena?
So what we've seen is it's an emerging area, and I think it's a very exciting area. And what we've seen is that the ability, in particular, to resolve full-length transcripts, also for single cells, is very exciting. There's a paper from, I would say, maybe 8 or 9 months ago from the Sanger Institute, where they demonstrate that it is possible to get information about the genome and the transcriptome from the same single cell. And in this particular study, PEG biosequencing was used again in this complementary fashion that I mentioned at the end of my talk, to not only get counts, but also get isoforms, different transcript isoforms for this particular single cell. These were in cancer cells. And so I think it's a first demonstration that I know of where the technology can also be useful for highlighting differential splicing and changes in the transcriptome architecture at the level of single cells.
Okay, we'll give you another few moments to send in questions, which you can send them via the green Q&A button at the lower left of the presentation window. Do you have any more questions? Okay, transcription, let me just see. I think it would be, is transcription affected by environmental factors? So how do you evaluate or control those factors in sequencing data?
So I would say that this is probably done most prominently through the study design. The sequencing will give you the underlying information about the genome, the transcriptome, and certainly the environmental factors, I think, are also known to affect the epigenome. And we didn't have time today to talk about the unique capability of PEG biosequencing to characterize the methylation status of the DNA as a process of sequencing. But it is certainly true that if you design your experiment properly and have cases and controls for the different environmental factors, then the sequencing can give you information about how these environmental factors change either the genome. Bobby Sebra at Mount Sinai presented a study at AGBT that was recorded. So if you go to our website and search for the AGBT workshop, he gave an example where he irradiated cancer cells and then with the sequel system and through PEG biosequencing looked at the changes in the structural breakpoints at particular genes, in particular the RB1 gene, and identified new structural breakpoints and hotspots in breakpoints through this irradiation of the cancer cells. And so this is true for the genome level, the transcriptome level, and the epigenome level. So with a properly designed study, the sequencing data can give you very important information on how that changes the genomic constitution of the samples.
Let's see. What is your opinion about the future sequence methods that can be used?
So I would say that at a general level, at a high level, we've seen a transition from sequencing being sequencing, and by that I mean that before the next generation sequencing and the third generation sequencing technologies came along, there was really only one method, and that was Sanger sequencing. And the research community had to adapt their particular research projects to what Sanger sequencing could provide. This is no longer the case. We now have quite a wide variety of different sequencing technologies, and they have different strengths and weaknesses. And I was trying to point those out during my presentation, that the Illumina technology is extremely good for counting because you get lots and lots of reads. And so you can count whatever's in your sample really well. However, there are limitations with regard to read length, with regard to bias, so that other applications like de novo assembly, like resolving some of these repeat expansion diseases, like resolving full-length transcripts, is not very well suited with short-read sequencing. And so that's where PacBio has a strength. And of course, on the flip side, we don't have the billions of reads, and so it's not a very good counting tool. So what that means is that the researchers now have choices, and depending on what they're trying to achieve and the questions they're trying to address, they have different choices of the different sequencing technologies. And so, you know, our job is to try to help guide the community to apply the best technology to the particular needs. Now, long term, I would say that when long reads become as cheap and plentiful, so when the technology will get scaled up so that it is possible to get billions of reads through smart sequencing, you know, that'll take some time, but I think fundamentally that will be possible. Then I think ultimately people will move to long read sequencing technologies as the default for everything, because then there's really no remaining advantage. The main advantage is the counting and the low cost right now. And you know, there was a series of Mendel's Pod interviews last year where the moderator, Thurald Timson, asked 5 key opinion leaders, 5 leading scientists in their particular area. He asked, if long reads become as cheap and as easy to get as short reads, would anybody still do short reads? And they all said no. So I think long term, that's where the field will move, and we'll see a reunification of sequencing technologies for many areas, because we're going to have a technology that provides the best possible performance in all areas that are important for a particular research project.
The next person says, sorry for the beginner level questions. I'm an undergrad trying to expand my knowledge in the field of genetics. So the question is, how are epigenetic modifications detected by sequencing?
Yeah, I apologize, I didn't get to that part. And the way it's detected is through analyzing the kinetics of polymerization. So as the polymerase moves along in real time and makes the complementary DNA strand, if that polymerase encounters a methylated base on the template strand, it slows down temporarily. So the methyl group acts like a little speed bump to the polymerase, and we measure that delay. And then through a statistical t-test, we can determine that that particular base was methylated.
When do you think SQL will be optimized to be able to sequence a human with comparable RS2 metrics?
So as you know, or as you may be aware, we have continuously improved you know, on the RS2 over the years, and that will also be the case for SQL. So as I presented in at the AGBT conference, some SQL data, we're now getting around 5 gigabases of data with an average read length that's also on the order of 10,000 bases. And you know, it also is very sample dependent and DNA quality dependent. So the read lengths might still be a little bit shorter than it is true now for the RS2, but you know, this will all you know, improve over time. And so I want to point out that these improvements are happening on a continuous level, and so on the same hardware over time, the overall throughput will improve, and we expect that to go far beyond the 5 gigabase level. And then I mentioned, of course, in the talk that there will also be the opportunity to increase the physical multiplex. So this will all happen in a continuous manner, you know, over the next couple of months, and then more long term, we'll continue to improve the technology over the next few years.
I would like to once again thank Dr. Korlach for his presentation. Do you have any final comments today?
Thank you, Judy, and thanks everyone for attending. I guess my final comment is that please feel free to contact us if you have further interest. We didn't get a chance to address all of the questions, so I know that they can be passed along, and I'd be happy to answer those to you directly. And check out our website, www.pacb.com, and you can reach out there, and we'd be delighted to hear from you and engage in a discussion to see how you can consider PEG biosequencing for your research projects. Thank you very much.
Thanks to the audience also for their great participation. This is an educational webinar and thus offers free continuing education credits. After the webinar is over and to get your credits, click on the CE button located in the bottom left-hand corner of your web page. Today's webcast will be available for on-demand viewing through November 11th, 2016. You'll receive an email from us alerting you when it's available on demand and posted on labroots.com. You're welcome to forward this announcement to any colleagues who weren't able to join in today. Thanks for logging on and participating in today's broadcast. See you next time.